Hi, we're going to present our comprehension activity 2. Our teacher is Ricardo León Acosta. We are from group 302, and the team members are Brandon Alberto Olivares Treviño, Sofía Berlanga Dávila, Octavio Guayo Mancha, Valerio Osornio Padrón, Jacqueline Villagrán Guillén, and me, Marcelo Sánchez Ríos. So we are going to present photosynthesis, cellular respiration, and fermentation.
What is the photosynthesis? The process is carried out by plants, algae, and some types of bacteria which capture energy from sunlight to produce oxygen, and chemical energy is stored in glucose. The plant then releases the oxygen back into the air and stores energy between the glucose molecules.
Steps of photosynthesis. Step 1. To start photosynthesis, leaves open their pores. This is also known as stomata. Step 2. Sunlight enters the leaves through the open pores. Step 3. Leaves take carbon dioxide, water, and minerals throughout the air and ground. Step 4. The plant converts water and carbon dioxide with the help of energy into glucose and oxygen. Step 5. Glucose will change the plant green color. This pigment is known as chlorophyll. Step 6. Finally, this process releases oxygen into the atmosphere. The enzymes for the photosynthesis. In the Calvin cycle, ATP and NADP are used to reduce or fix carbon dioxide to make glucose. This carbon fixation reaction is catalyzed by an enzyme called rubisco, a highly abundant protein in plants.
During photosynthesis, 18 molecules of ATP are used in C3 plants. Out of these, 12 are used in synthesis of 1-glucose molecules and 6 for regeneration of RUBP. The light in photosynthesis. The light reaction occurs on the thylakoid membrane in the chloroplast inside the plant cell. They get underway when light strikes a protein chlorophyll complex called the photosystem II. This enzyme is what liberates the hydrogen atoms from water molecules.
Cellular respiration. What is it? Cellular respiration is a three-step of metabolic reaction and process that take place in the cells of organisms to convert chemical energy from oxygen molecules or nutrients into adenosine triphosphate, ATP, and then release waste products. This is carried out through metabolism, which is any series of reactions either of synthesis or degradation of molecules within a cell. Aerobic respiration is carried out in the presence of oxygen and can produce a lot of energy, while anaerobic respiration takes place in the absence of oxygen. Catabolism creates energy for cells from degrading organic nutrients, while anabolism produces new components absorbing the energy applied to the functioning on the cell.
The steps of catabolism. First, macromolecules in food are transformed into smaller molecules. This stage acts on starch, cellulose, or proteins that cannot be directly absorbed by cells. Second, the molecules from the first stage are transformed into acetyl-CoA, which releases some energy. And third, acetyl-CoA enters the Krebs cycle. The reducing equivalents that are used in the electron transport chain are generated. This step creates at the end water. Steps of cellular respiration. Step 1. In glycosis, that is the pathway that the glucose catabolism performs, undergoes a series of chemical transformations. In this reaction, ATP is made and AD is converted into NADH. Step 2. Each pyruvate from glycosis goes into the mitochondrial matrix. It's converted into two carbon molecule bound to acetyl-CoA, then carbon dioxide is released and creates NADH. Step 3. The acetyl-CoA combines with 4 carbon molecule and goes through a cycle of reactions. ATP, NADH, and FADH2 are produced and carbon dioxide is released. Step 4. As the electrons move down the cycle, energy is released and used to pump protons into the matrix through ATP synthesis, making ATP. At the end of the electron transport chain, oxygen accepts electrons and takes up protons to form water.
What is fermentation? Fermentation is a chemical process in which molecules are broken down anaerobically. Anaerobic breakdown was discovered in 1920. The absence of air extracts of muscle catalyze the formation of lactate from glucose, and that the same intermediate compounds formed in the fermentation of grain are produced by muscle. Types of fermentation. Lactic acid fermentation. Yeast strains and bacteria converts starches or sugars into lactic acid, requiring no heat in preparation. Ethanol or alcohol fermentation. Yeasts break pyruvate molecules, the output of the metabolism of glucose, known as glycolysis. It in starches or sugars down in alcohol and carbon dioxide molecules. Alcoholic fermentation produces wine and beer. Acetic acid fermentation. Starches and sugars from grain and fruit ferment into sour-tasting vinegar and condiments. Examples include apple cider vinegar and wine vinegar.
Steps of fermentation. Primary fermentation. In this brief phase, microbes begin rapidly working on raw ingredients such as fruit, vegetables, or dairy. The microbes present in the surrounding liquid, such as brine for fermented vegetables, prevent putrefying bacteria from colonizing the food instead. Yeasts of the other microbes convert carbohydrates, sugars, into the other substances such as alcohols and acids. In secondary fermentation, the longer side of fermentation lasts several days or even weeks. Alcohol levels rise and yeasts and microbes die off, and their available food source, the carbohydrates, becomes scarcer. Winemakers and brewers use secondary fermentation to create their alcoholic beverages. The pH of the ferment can differ significantly from when it started out, which affects the chemical reactions taking place between the microbes and the environment. Once alcohol is between 12 and 15% and it kills the yeast, preventing further fermentation, distillation is needed to remove the water, condensing alcohol content to create a higher percentage of alcohol. Thank you for your attention.